Hi, and welcome to the Membrane Dynamics lecture. This is going to be a pretty in-depth lecture, and it is 50 slides, but it's only because I've broken down all the mechanisms into separate slides that give you a step-by-step. So in reality, it's really should have been only like 30 slides if I had condensed it onto one page. But to make it easier and to simplify the mechanism steps, I've put them each on separate slides. So let's get started. We have to cover some ground material first before I cover the mechanisms, and a lot of this should be review from lectures 1 and 2 that we went over. But in your body, you have different compartments, like the cell. The inside of the cell is called intracellular fluid. The outside of the cell is called extracellular fluid, also known as interstitial fluid. In your blood vessels, you have blood cells, and then the fluid around them that is called plasma. We're really going to be focusing on intracellular fluid and the interstitial fluid today. Now, this slide should be pretty much a review because we've gone over the salt composition of intracellular fluid and interstitial fluid, also known as extracellular fluid. So, intracellular fluid is in the cell. You have a high concentration of potassium and proteins. Outside of the cell, the interstitial fluid, also called extracellular fluid, has a high concentration of sodium and chloride. Why are these salt concentrations important? Well, because it dictates how solutes move based off of diffusion. Remember, the definition of diffusion states that solutes always want to go from areas of high concentration to low concentration voluntarily. So here we have the definitions once again. Osmosis is the movement of water, and that's going to be water moving towards areas of high solute concentration. And the movement of water generates what we call osmotic pressure, and we'll look at this in just a moment. We have the terms molarity versus osmolarity, which we will define as well. And we have the terms isoosmotic, hyperosmotic, and hypoosmotic. The next few slides will cover these 4 bullet points and will all pertain to osmosis. Slide number 1 here about osmosis is about the definition of osmosis as well as osmotic pressure. Here you have a U-tube that is separated by a semi-permeable membrane. This just means that the membrane is partially permeable, and we've decided that we're only going to allow it to be permeable to just water. Compartment A has a lower concentration than compartment B. I know that because I can see that there's a lot more glucose molecules in compartment B. Another way is to say, hey, if you took a straw and you tasted the water here versus here, which one would have a stronger flavor? Well, B would taste a lot more sweet. Now, by definition of osmosis, again, water wants to move towards an area of higher concentration. We just said that compartment B has a higher concentration. So with that being said, the water from A is going to want to move over to water in B. If you can see the concentration changes now, it looks like if you were to take a sip of water from here versus a sip of water from here, the flavors or the sweetness of it would probably be equal. Osmosis took place to make sure that the distribution of concentrations was equivalent. So here's where I'm going to give you a statement, and you should write this down. Water will always follow salts or solutes. Wherever there is a lot of salt or solute, the water will follow. Now here is where we start talking about osmotic pressure. Pretend you had a force applied to side B. You had a little rubber stopper put into the side B of the U-tube. This rubber stopper would have to push down with enough force to prevent osmosis from taking place. The amount of pressure that you have to apply to resist osmosis from happening is called the osmotic pressure. It is the pressure required to oppose the movement of water. Because again, remember, water wants to go from compartment A to compartment B. But if you force the top shut and you don't allow any more filling, that pressure applied is the osmotic pressure. Now, in terms of describing concentrations of compartments, we have 3 terms to describe concentrations of compartments in relation to one another. So let's say we had 3 compartments. Compartment A was 1 osmol glucose. Osmol is how you measure osmolarity, which is the concentration of solute in water. Solution B is 2 osmols of glucose, and solution C is again 1 osmol of salt. So osmolarity is the way you measure the solute concentration within water. 1 is less than 2, 2 is more than 1, and then A and C are equal to each other. There are 3 terms we're going to use to describe the relationships between these concentrations: hypoosmotic, hyperosmotic, and isoosmotic. Now, before I give you these concentration definitions, I need you to write this down. Anytime you see the prefix hypo, it means low. Anytime you see the prefix hyper, it means high. Anytime you see the prefix iso, it means the same. So what this means is we can use these terms to describe concentration relationships. For example, hypoosmotic means lower in concentration. So a statement I could make is that A is hypoosmotic to B. That means A is lower in concentration to B, 1 compared to 2. Or I could say B is hyperosmotic to A. I could also say B is hyperosmotic to C because B is higher concentration than A or C. Then when using isoosmotic, I could say A is isoosmotic to C because they're the same concentration. So these terms are used to describe 2 liquids in relationship to one another in terms of their osmolarity. Now, slightly moving on to diffusion. This is one of the most important concepts you will learn right now in this first chunk of material. You have to understand diffusion to understand the rest of this class. The definition of diffusion is right over here in number 2. It's movement of solute from high concentration to low concentration. We call this movement moving down a chemical gradient. So solutes always want to bounce around from areas that there's a lot of them towards areas where there's not so much of them. This is a very passive process. This means that it does not require energy or ATP. And diffusion will take place until those solutes are spread out evenly and they're in equilibrium. Diffusion happens very fast over very short distances, or can take longer over longer distances. Diffusion is directly related to temperature, so the warmer it gets, the faster the molecules move, the faster they can diffuse. They are inversely related to weight and size, which means the heavier a molecule is, the longer it's going to take for them to move around and diffuse. And diffusion can happen either in an open system, which means let's say I spray Febreze on the corner of one big open room. It'll move eventually to the other corner of the room. We call that an open system. Or across a partition. Let's say, and I'm going to draw this out for you, we have a compartment that looks like this, and in the middle, it is separated by a barrier. And this barrier has little tunnels that allow for the movement of that Febreze. If I spray the Febreze over here, it will eventually go through the openings of the partition and move to this compartment. So that's what we mean by partition. It can happen through little tiny holes or through just an open space. So here's an example of diffusion that's based off of mass. So you have a petri dish that has either potassium iodide in it or Congo red. This is much heavier than potassium iodide. This guy's a little baby molecule. He's a really big Mongo molecule. If we put these solutions on an agarose that's on a Petri dish, they're going to diffuse out. Now, what dictates how fast or how far they diffuse? Well, temperature was one of them, but the big one here is going to be molecular size. Because potassium iodide is much smaller, it diffuses much farther out, versus the Congo red has a much smaller radius of diffusion because it was much heavier. Now, rates of diffusion across membranes are affected by a few different things as well. I'm mentioning this because really we're going to talk about diffusion across membranes only for the remainder of this PowerPoint. So here's a lipid bilayer, and you have some things that can affect your rate of diffusion. Molecular size. The smaller it is, the easier time it's going to have getting through the membrane. The bigger it is, the higher the chance that it's going to bounce off the membrane. Then lipid solubility. Because the membrane is made mostly of lipids, like in this tail region here, if the solute is a lipid, it's going to be able to get through a membrane that's also mostly a lipid. However, if it is not lipid-based, it's most likely going to go pew and bounce off. The membrane surface area, the more surface area you have, the more chance a solute has to actually cross through. And then the composition of the lipid bilayer. If you recall, I believe it was in like the second or third PowerPoint, we talked about how this membrane has proteins in it. It has cholesterols in it as well. Depending on how many proteins you have, how many cholesterols you have, that will also affect the rate of diffusion. So because lipid bilayers, if I can just show you again, they for the most part are just lipids that are put together. They are mostly hydrophobic. The chances of molecules actually going from the outside of the cell into the cell, where let's just say sugar would need to go in to get broken down into energy for you, it would be very difficult to do that across a lipid bilayer. But let's say I put in a big protein transporter that transported sugars. Then, let's draw a really big arrow, I would be able to bring in a lot more sugar. So because of this, we like to have transporters and channels. Channels, if this was your membrane, is going to be just an open doorway that can either have a gate on it or just be an open doorway for molecules to get through. Channels are like tunnels. You have channels for water, you have channels for different ions like sodium, potassium, and chloride. And you have channels that can be open channels that can just remain open, or gated channels that can be opened or closed depending on chemicals that are present, the voltage of the cell, or physical change in structure of the channel. We're going to cover a good number of these, and I will give you examples of most of them. So here's the illustration of it. Your membrane transporters, those are proteins in the membrane that help you to transport things into or out of the cell. You have either 1, channel proteins, or 2, carrier proteins. Channel proteins are the ones we just talked about. They're little tunnels in the cell membrane. They allow for things to move in and out. This is 100% dependent on diffusion. Write that down. Channel proteins move solutes down their concentration gradients only. Now, channels can either be open channels where they just stay open, which are not a whole lot of them, or they can be gated channels, which are predominantly the types of channels we're going to see. Gated channels can either be in the open formation or the closed formation. What decides what formation it's in? Well, again, chemicals that are present, voltage that's present in the cell, or a physical change in the structure. We'll talk about these a little bit more in a few slides. Carrier proteins, however, if you notice, they're first open in one direction, and then they close up like this, and then they open in the opposite direction. These are carrier proteins. They can use ATP, they don't use ATP. Well, what is it dependent on? Again, if the solute is moving down its concentration gradient, like from an area of high concentration to low concentration, it's not going to require ATP. However, if you're moving things against their gradient, like sodium, you're moving sodium from an area where there's very few sodium to an area where there's already a lot of sodium, you're moving it against where it naturally, by diffusion, wants to go. Same with potassium. You're moving it towards an area where there's already a lot of potassium. You're moving these guys against their gradients. At that point, it requires ATP. You have to force them with energy. So with that being said, we'll cover that more in just a moment. I do want to go over these words: uniport carrier, symport carrier, and antiport carrier. Uniport carrier, uni means one. That means it's moving one thing in one direction. Symport carrier, sim means same. So you're moving two things in the same direction. Or antiport carrier, anti means opposite. You're moving two things in the opposite direction. All of these carriers are in this structure. Although it's not shown in this cartoon, just in your head, know that they open at one end, close up, and then open up at the other end. So with that being said, let's look at the formation of this carrier. Let's say you want to move a solute from the outside of the cell to the inside of the cell. The solute comes, finds its binding site. Once it binds here, that is the signal to tell the carrier to close. And then it opens up to the inside of the cell and it tells the solute to leave and go into the cell. Now this illustration doesn't show you anything about ATP or energy usage. This is just showing you the orientation of carriers. So they're open at one end, then they shut on both ends, and they open up to the opposite side. Now, we'll start with our first big mechanism. And I'm going to start by talking to you about one particular transporter. It is the king of all transporters. You will see this transporter in every lecture from this point forward until you're done with this class. Why? Is it because I want to show it to you? Well, I do like to show it to you most times because it's very helpful to see it. But also, I can't teach you a mechanism without actually showing you this transporter and that mechanism. This transporter, called the sodium-potassium pump, is found in every single cell of your existence. It happens 1 billion times a minute, and you would die without it. The sodium-potassium transporter, like you probably guessed, moves sodium and potassium. These are called active transporters, not passive, but active. If passive transporters do not require energy, what do you think an active transporter is going to require? ATP. It requires energy in the form of ATP. And if you recall from the last two slides, you will always need ATP when you are moving solutes against their concentration gradient. So if I have a lot of sodium here and a very little bit of sodium here, normally, by diffusion, sodium wants to go in this direction. That's by diffusion. Passive, that's the desirable direction. But what if I want to take this sodium and move it to this area? Is it going to voluntarily want to go from low concentration to high concentration? Well, no, that's not the definition of diffusion. But I want it to go in this direction. So what am I going to have to apply? Or what am I going to require to force sodium from here to go to here? Well, I'm going to require ATP, and that's known as an active process. So here we go. We're going to talk about the sodium-potassium pump. We're going to start by just looking at the concentration gradients in and out of a cell. This is something you should know now. We've been over this probably 4 times now. Inside the cell, which ion do you see a lot of? sodium or potassium? We've got 1, 2, 3 sodiums. We've got a lot of potassiums. So inside the cell is a whole lot of potassium. Outside the cell is a whole lot of sodium. So naturally, all of this sodium says, ooh, I want to go into the cell. And all this potassium is like, oh my gosh, I want to leave the cell because there's not a whole lot of me out here. But the sodium-potassium pump, this transporter here, with the use of ATP, is going to force sodium to leave the cell against its gradient, and it's going to force potassium to enter the cell against its gradient. Now you might be wondering, why are we forcing things to go where they don't want to go? Well, to maintain a proper salt balance inside and outside the cell, you have to make sure you maintain these concentration gradients. We will talk about why this is, I believe, at the end of this PowerPoint. But for now, we have to keep sodium high on the outside and potassium high on the inside. So how does this pump work? So this pump is like a transporter, and it starts off with step 1. Open that transporter to the intracellular fluid inside the cell. The sodium-potassium pump has 3 binding sites for sodium and 2 binding sites for potassium. This is just simply the structure of a sodium-potassium pump. So when this transporter is opened up into the cell, it's going to bind 3 sodiums from in the cell into the 3 binding sites in the transporter. Now with the use of ATP, we're going to force that transporter shut. And when I say use ATP, we're going to take adenosine triphosphate, pluck a phosphate off of it, bind it to the transporter because that's where you have your energy, and it turns into adenosine diphosphate. And this will just be a byproduct. It'll go into the cell. So with the use of ATP, we forcibly shut the transporter. Our energy-full phosphate is tagged onto it, and now we can take this transporter and open it up to the outside. So again, with the use of the ATP, I was able to shut the transporter forcibly and open it to the outside of the cell. Because of this structural change, these binding sites kicked all of the sodiums out, and those sodiums are going to go out of the cell. At this point, you've now successfully taken sodium from the inside of the cell and kicked them out of the cell. It will always be 3 sodiums. Make sure you know that number. At this point, you now have the availability of the potassium binding sites there and there. 2 potassiums, and that is 2, are going to come in and come into their binding sites. Once they bind into their binding sites, the phosphate will be released out. And because we had a change in structure, remember, even just releasing 1 phosphate will cause a domino effect of structural change. That release of the phosphate and the binding of the 2 new potassiums causes this transporter to shut on one side and open up on the intracellular side, releasing the 2 potassiums into the cell. So looking at all of this again, you'll notice that this transporter moves 3 sodiums out of a cell, and it moves 2 potassiums into the cell. It is a 3-to-2 ratio. Make sure you know this ratio as well. So this is where we talk about why it's so important to make sure you have high sodium on the outside of a cell and high potassium on the inside of a cell. You have to constantly keep high sodium on the outside, because if you do, sodium will constantly have a high to low concentration gradient. You want to maintain sodium's high to low concentration gradient, remember, by definition of diffusion, a passive process that allows solutes to move down their concentration gradients is what we're looking for. Why is that? Because sometimes there is another molecule, and we'll use an example of glucose, and I'll just write Gl. Glucose must always enter the cell to be broken down into energy. Without glucose going into your cell won't have energy, and your cell will die. Now, here's the issue. You already have a lot of glucose in the cell. Now, the intracellular glucose concentration is much higher than the extracellular glucose concentration. This is how I'm symbolizing high concentration and low concentration. Is glucose going to want to enter the cell to a place where there's already a lot of it? No, that is not the definition of diffusion. So what is it going to require to force glucose into the cell? ATP. But you don't want to keep using ATP to get glucose into the cell just to make ATP. Seems kind of counterintuitive. So I want to figure out a backdoor kind of undercover method of sneaking glucose into the cell without using ATP. So what I'm going to do is I'm going to piggyback him. So glucose says, hey, sodium, I see that you're getting into the cell for free. How are you getting in the cell for free? Well, sodium says, hey, I'm going down my concentration gradient. And glucose says, well, I don't have a downward concentration gradient. I'm forcibly going up my gradient to get into the cell. So glucose says, hey, sodium, do you think I can just tag along with you and use your downward sliding concentration gradient to get into the cell? Can you sneak me into the cell? Sodium says, sure, buddy, not a problem. So glucose tags along with sodium and kind of hides behind him, and they both get into the cell for free by using sodium's downward gradient. Now, this whole process is called a symport process. Remember, symport carriers move two things in the same direction. Glucose is not the only solute that does this. Chloride, bicarbonate, amino acids, protons, calciums, all of these essential solutes will use this type of symport carrier to move into the cell. We call this secondary active transport. Active because it's supposed to use ATP, but secondary because it doesn't actually use ATP. We piggyback it with a sodium to move down the concentration gradient. Now we're going to go through the mechanism in detail. Same one that I just taught you, the second time with cartoons. So again, we have sodium, high concentration outside the cell, low concentration inside the cell, so sodium has no problem diffusing it. But then we got glucose, low concentration outside the cell, and it already has a high concentration inside the cell. So is glucose going to voluntarily just diffuse into the cell where there's already a lot of him? Probably not. So instead of using ATP, glucose asks sodium, hey, can I piggyback with you and we can go in together? Sodium says, sure, no problem, buddy. So they go in through what's called the sodium glucose transporter. That is the name of this mechanism we're about to cover. So step 1, sodium goes into his binding site. Step 2, glucose will then go into his binding site. Now I want you to notice, this binding site here wasn't open initially. It only opens up once sodium binds in. Remember, when sodium binds into the protein, it changes the structure and changes the function. So sodium binds in, domino effect takes over, it exposes a glucose binding site. The minute glucose binds in, again, you change the structure and you change the function. It shuts the carrier on the extracellular side opens up on the intracellular side, allowing both solutes to enter the cell. The step-by-step is written here. Be sure you know the mechanism of the sodium glucose transporter. This is also called the sodium glucose symporter. Both names are valid. They both work. PhysioX might use the word symporter, but I believe on your test I put SGLT. Now, we have something called carrier-mediated transport. Carrier-mediated transport is very specific to specific solutes, and there can be competition for binding sites. What do I mean by this? Specificity is like something in this illustration. You have something called a GLUT transporter. This is called the glucose transporter. What is the only thing this guy can transport? Glucose. Very specific. So glucose binds in and it's able to be moved across a membrane. However, sometimes you have dimers of glucose. So when two glucoses come together and they make a bond, this is a new sugar called maltose. Maltose is like an artificial sugar. It's what we see in artificial sweeteners sometimes. We consider maltose a competitive inhibitor to the GLUT transporter. Now just break this word down for yourself for a second. Competitive, because it competes with glucose for this binding site, and an inhibitor, because it's inhibiting glucose from actually binding itself. When maltose binds, it's able to bind, but this transporter can no longer close, and it stops the function of the transporter. Hence, a competitive inhibitor. Then we have some terms here. This should be a review from biology. Phagocytosis is when a cell is able to engulf another particle or cell internally to digest it. This is an example of phagocytosis here. You also have something called endocytosis and exocytosis. Endocytosis is when you have a cell and you have a little tiny food particle. And as that particle gets closer and closer, it's able to actually be engulfed into the cell using a similar mechanism. It just does it for much smaller little solutes, not full bacteria or full cells. Exocytosis is when the cell actually spits out something from the inside to the outside. So it's going outwards, whereas this one is going inwards. So endocytosis brings it in. Exocytosis exits it out. The next mechanism actually has to do with this. We're going to go over receptor-mediated endocytosis. So I'm going to show you how a cell picks up something from the extracellular fluid, binds to it, and brings it into the cell.